Welcome back. I'm glad you could join me as we continue this journey into biology, a subject that quietly reveals just how intricate, ordered, and astonishing life really is. And as always, this lesson is brought to you exclusively by E-studylab as part of a course designed to guide you steadily, one idea at a time. In our previous session, we explored inheritance as something you can observe: height, eye color, skin tone, even family traits that seem to echo through generations. Today, we move one layer deeper. Instead of asking, what is inherited, we begin asking, how? Let's begin with the most important molecule in biology, DNA. DNA is often described as the blueprint of life, and that metaphor truly fits. Just as an architect's drawings tell builders how to construct a house, DNA carries instructions that tell cells how to build and maintain a living organism. Every detail, from your hair texture to the way your organs develop, begins as information stored in DNA. Inside each of your cells, DNA is tightly packed into structures called chromosomes. Along these chromosomes are short stretches known as genes, and each gene carries instructions for a specific trait or function. You can imagine DNA as a long sentence written in a 4-letter code. Those letters are chemical bases: adenine, thymine, cytosine, and guanine, and they pair in a precise way, forming a twisted ladder shape known as a double helix. The shape may be beautiful, but the purpose is practical. It allows DNA to copy itself with remarkable accuracy every time a cell divides. Now, DNA does not work alone. To turn information into action, DNA relies on another molecule, RNA. While DNA stays safely inside the nucleus like a master record, RNA acts more like a messenger. It copies instructions from DNA and carries them out into the cell where proteins are built. There are 3 main types of RNA working quietly in the background of every living cell. Messenger RNA carries the genetic message from the nucleus to a structure called the ribosome. Transfer RNA brings the raw materials, amino acids, to that ribosome, like deliveries to a factory. Ribosomal RNA forms the factory itself, the place where proteins are assembled. And what are proteins? They are the workhorses of the body. Proteins build tissue. They transport oxygen. They repair damage. They regulate chemical functions. They even allow muscle to move and the immune system to fight disease. In many ways, proteins are life in action, and DNA is the instruction manual behind them. Now let's shift from molecules to something that can feel more personal: difference. Why don't all human beings look the same? Why does one sibling inherit curly hair while another doesn't? The answer lies in genetic variation. Every organism carries a slightly different version of the genetic code. That variation is what gives rise to individuality, adaptation, and evolution. It is also what makes populations resilient rather than fragile. Variation comes from several sources. Sometimes DNA changes slightly when it copies itself. This is called mutation. Most mutations have no effect, but occasionally one introduces a new trait. Variation also arises through recombination, which happens during reproduction, when genetic material from both parents mixes to form a new combination that has never existed before. Populations also change through gene flow, when individuals move between groups and bring new traits with them. And sometimes changes happen by chance, a process known as genetic drift, where a trait becomes common not because it is useful, but simply because of random survival. Together, these forces prevent life from standing still. They ensure movement, adaptation, and diversity. Now we arrive at one of the most powerful and most debated developments in modern biology: genetic engineering. Genetic engineering is the deliberate changing of an organism's DNA using scientific techniques. Instead of waiting for traits to appear naturally, scientists can now edit genetic code directly. One of the most revolutionary tools in this field is CRISPR-Cas9. You can think of CRISPR as a pair of molecular scissors, able to locate a specific section of DNA and cut it, remove it, or replace it. This makes it possible to correct faulty genes, insert new traits, or alter organisms in precise ways that were once unimaginable. Through genetic engineering, crops can be made more resistant to disease. Insulin can be produced for diabetics. Experimental treatments can target inherited illness at its source. But with power comes responsibility. Genetic engineering raises serious ethical questions. Should we edit human DNA? Who decides what counts as improvement? Could new technologies increase inequality between people? And what are the risks of altering ecosystems by changing the genetic structure of plants or animals? Biology at this level is no longer just a science. It becomes a conversation about responsibility, fairness, and the future of human life. In the coming weeks, you'll meet biotechnology again as we explore microorganisms and medical science. For now, hold on to this simple truth. Life is not only built from cells, it is written in code. And understanding that code opens a door not only to knowledge, but to wisdom. Before we meet again, you might enjoy noticing heredity at work in your own world, family features, inherited talents, or even how plants resemble their parents. Biology is never far away. A quiet but important reminder that this lesson is protected by copyright and is owned by E-studylab. When you're ready, we'll step beyond genes and into ecosystems, where living things interact with one another and with the world that holds them all.